Vaccines are used to develop immunity. In this part, we are going to learn vaccines, vaccination, and a small topic, hypersensitivity. Dr. Edward Jenner prepared the first vaccine for smallpox in 1796. A microbiologist, Louis Pasteur, in 1885 discovered vaccine against rabies, anthrax, and cholera. Using vaccines to prevent and control diseases has been one of the greatest achievements in the history of medicine. According to WHO, after the development of vaccines, the death due to infectious diseases has been reduced greatly. So vaccines development played a significant role in reducing the death due to infectious diseases. A vaccine is a biological preparation. Vaccine contains biological material. Vaccine provides active acquired immunity. A particular vaccine provides immunity against a specific disease. Vaccine resembles a disease-causing microorganism. Actually, a disease-causing microorganism stimulates immune response. What is immune response? Antibody production is an immune response. Activation of cell-mediated immunity is an immune response. The vaccine also promotes immune response like a disease-causing microorganism. A vaccine is made from weakened or attenuated microbes. Microorganism is weakened, so its ability to cause disease is stopped. Or killed form of microbes are used, or toxins of microbes are used in vaccines. In some cases, one of the microbes' surface proteins are used. The surface proteins which act as antigens are used. Recently, after 1990, DNA vaccine becomes produced. DNA vaccine development took place after 1990. Now let us see what do the vaccines do. The vaccines develop immunity. The vaccines stimulate immune response. That immune response is in the memory of the immune system. When viruses or bacteria invade our body, these vaccines teach our body how to protect itself. A vaccine delivers a small number of microorganisms. Very little amount of inactivated or weakened viruses or bacteria are introduced into the body. This small amount of weakened microbes allow the immune system to recognize the organism. Our immune system recognizes the organism and develops immune response without actually experiencing the disease. Without developing a disease, the immune system recognizes the antigen, that is the microorganism which is injected during vaccination, and develops immunity. In some cases, the vaccine has to be repeated, that is booster vaccination. Some vaccines need to be given more than once. Such vaccines are called booster vaccination. Based on the nature of the preparation, there are 3 generations of vaccines. The first generation vaccine is a traditional type. It includes live attenuated vaccine, killed vaccine, toxoids. Weakened, attenuated, aged, less virulent form of the viruses are used in attenuated vaccines. Here the microorganisms are not killed. but they are weakened by a specific culture method, or aged cultures are used, or less virulent form of viruses are used. Here the example is MMR vaccine, measles, mumps and rubella vaccine. Another example is varicella vaccine. It provides protection against chickenpox. Killed vaccines are safe. Here the microorganisms are killed or inactivated by heat, and other methods are used, but here there may be a need for the booster vaccination. Example, Salk's polio vaccine. Here the Salk's is the name of the person. Toxoids are vaccine containing toxins or other chemicals produced by bacteria or viruses. Example, DPT vaccine, diphtheria, pertussis and tetanus. So these three types come under first generation vaccine. They are sometimes called as traditional vaccines. The second generation vaccine contains the surface antigen of the pathogen. The surface molecules which act as antigen are used in the vaccine preparation. Example, hepatitis B vaccine. Third generation vaccine, plasmid containing a gene is introduced. The vaccination introduces a plasmid carrying a gene into the body. The plasmid carries the gene into the cell, and inside the cell the gene encodes the antigen. The gene causes the production of antigen. So antigen is produced inside by the host cell. Plasmid carries the gene into the cell. Inside the cell, the antigen is produced by the host cell. So immune response of the body is stimulated by a DNA molecule. Here the vaccine introduces a gene into the host cell. That gene carries information for an antigen. So this antigen is produced by the host cell itself. Here immune response is stimulated by DNA molecule, that is a gene. So sometimes this vaccine is called as DNA vaccine or recombinant vaccine. We will discuss about this vaccine in the chapter 9. Vaccination is the process of introduction of vaccine into the body to produce immunity to a particular disease. Vaccine is introduced to develop immunity to a particular disease. The first vaccine given to newborn babies, BCG. This vaccine is introduced to develop resistance against tuberculosis. This is vaccination. And after vaccination, the body develops immunity, that is immunization. The development of immunity by the body after vaccination is immunization. A vaccine stimulates immune response, that is antibody production or activation of cell-mediated immunity. This immune response generated by vaccine is recorded in the memory of the immune system. So when the infection occurs for the next time, the immune system exhibits a fast, quick response, a powerful response. This is a mechanism of working of vaccine in developing immunity. The second topic we are going to discuss here is hypersensitivity. Hyper means more. Sometimes our immune system shows over response, excessive response against a common substance, that is hypersensitivity. Pollen particles in the garden cause sneezing, running nose in the sensitive individuals, and some kinds of food items cause allergy on the skin and develop skin rashes tears on the eyes, stomach problems, vomiting, skin rashes. Some medicines cause skin allergy. Nickel-like substances, chemicals cause skin allergy. So the immune system shows excessive response or inappropriate response against some common substances. The substances which cause excessive response of the immune system are allergens. Hypersensitivity is the exaggerated or excessive response of the immune system. This response of the immune system is caused by some substances. They are called as allergens. Example, dust particles, pollen, some proteins in insect venom. Honeybees and wasps, they have venom, and snake venom. Some chemicals cause allergy. Example of the conditions, hay fever and asthma. The symptoms of allergy includes skin rashes, sometimes vomiting, sneezing, watery eyes, running nose, and difficulty in breathing, and various other symptoms. Allergy is a form of overactive immune response mediated by IgE and mast cells. The mast cells and immunoglobulin E involved in developing these symptoms. Mast cells secrete chemicals. The mast cells secrete chemicals like histamine and serotonin. The histamine serotonin cause the allergic response. Sometimes the allergy causes a violent reaction, quick response, a fast reaction. This anaphylaxis reaction, fast reaction of the immune system, is due to the result of rapid generalized mast cell degranulation. Mast cells are secretory cells. They secrete histamine like chemicals. Here degranulation means development of secretory vesicles from the Golgi bodies. These vesicles secrete chemicals. So mast cells produce more chemicals, allergy chemicals, that results in the fast response. This will happen against some insect bite, snake bite, and some allergic individuals. The penicillin antibiotic develops anaphylactic reaction. In this part, we have covered two topics: hypersensitivity and vaccines. In the next part, we will discuss about immunodeficiency disease.